This is the course outline for this chapter. This chapter is divided into two: expressions of biological information 1 and expressions of biological information 2. For the second part, we will look at the 2.4 and 2.5. For the first part, we will go into three topics: DNA replications, and transcriptions and translations. So last week, I think you already have your lecture with Dr. Shafa. So that lecture, you learn, you discuss about Mendel law of inheritance, law of independent assortment, and law of segregations. So actually, before that, almost up to hundreds of years, nobody knows what is the factors that Mendel's refer as the alleles. So after that, there are many subsequent experiments that prove something inside the cells, something inside our cells that is responsible for the inheritance, the hair color, skin color, and everything. So from Mendel discovery, for hundreds of years, the scientists try to find what is the factors, and they refer the factors as genes. See, there are hundreds of years before the genes are proven to be located on the chromosomes. And then after James Watson and Francis Crick introduced the structure of DNA, deoxyribonucleic acid, then the expressions of biological information science is being explained. So we know that DNA is the molecules that is responsible for inheritance, is responsible for the characteristic of our characteristics. For example, our hair color, skin color, eye color, and everything. So from the Mendel discovery, we find the factors. The factors is located on the chromosomes, and in the chromosomes, there are these molecules, which is DNA. So DNA is the molecules that is responsible for everything. Okay, this is the timeline actually. From Mendel discovery, for hundreds of years, nobody knows what is the factors. The factors is called genes, and the genes is now discovered as DNA structure, inside the DNA structure. But the discovery actually is the results of few experiments, because in biology you cannot say this is DNA and everyone will accept the fact that this is DNA. In order to prove the DNA is the molecule that is responsible for the inheritance, you need to provide with proof. Proof can be provided using experimentations. So that's why you are a science student, you do experiments every day. Every semester you do several experiments. Actually, the experiment that you do in lab is a result of previous experiment. You just repeat the experiment so that to prove something that you learn in class, like this one. Theoretically, you learn something, you learn about DNA, but then what you learn actually is a result of several experiments. And the experiment begin with Frederick Griffith. They discover the genetic roles of DNA by using this experiment on mice. Okay. put two strains of bacterium, one pathogenic, one is non-pathogenic. When they put the pathogenic strains inside the mouse will die. On the other hand, if they put the non-pathogenic strain of bacteria into the mouse is still healthy because the bacteria, they don't have any disease that will infect the mouse. And then they do another experiment where they heated the pathogenic cells, bacterial cells, and they inject the same way with the first experiment. Yeah. The first experiment, the mouse died because the S cells is alive. But when they kill the S cells, the mouse is healthy. So that this means that the pathogenic bacteria just now, when they are heated, they cannot infect the mouse. But when the heated pathogenic bacteria is mixed with a live non-pathogenic bacteria, they undergo a process that we call transformation. They undergo transformations, which means that the heat kill S cells, the pathogenic cells, transform the non-pathogenic live cells into pathogenic. And now we know that the one that transforms the non-pathogenic cell is the DNA. Okay. Because when you heat kill the pathogenic cells, the cells will die, but the DNA remain. So when you mix the DNA with live cells, non-pathogenic bacteria, the DNA will go inside the non-pathogenic bacteria and they will transform the non-pathogenic bacteria into pathogenics. So that's why when you infect the mouse with the DNA of pathogenic bacteria, the mouse will die. Okay, don't worry if you cannot understand this experiment now, but along the way, you will understand the concept of DNA, how DNA works, and then you go back to this experiment, and you will have a better understanding about this topic. Don't worry, because this experiment is done by experts. So if you have difficulties to understand, it's okay. Just to show you that all the information in this lecture is based on experimentations by previous scientists. Okay, after the experiment by Frederick Griffith, Oswald Avery, McCarthy, and McLeod continue this experiment and show that the only DNA work in transforming harmless bacteria into pathogenic bacteria. Because in Frederick Griffith experiment, they heat kill the pathogenic bacteria. But when you heat kill the pathogenic bacteria, not just DNA is remain active. There are RNA, there are proteins and other macromolecules. So we don't know. At that time, they don't know which of the molecules is responsible for the infections on the mouse. So the Oswald Avery, Maclean McCarthy, and Colin McLeod, they continue the Frederick Griffith experiment with the objective of proving only DNA and not other macromolecules. So this is the experiment. They use two enzymes that will hydrolyze or they will remove RNA or DNA based on it. RNase is the enzyme that will remove RNA. DNase is the enzyme that will remove DNA. So when they treat the mixtures of the pathogenic bacteria just now, the mixtures, they treat with RNase, the RNA will be removed. So only DNA remains. So they add the mixtures to the bacteria. At this time, they didn't use mouse anymore. They just grow on the agar plate. So if they grow on the agar plate, meaning they are alive. So when the RNA is removed, the bacteria, the pathogenic bacteria just now, they can grow on the agar, meaning that they is alive and can infect the mouse. But when they treat with DNase, DNase will remove DNA. So only RNA remain in the mixture. So when they grow the mixture on the agar plate, the bacteria didn't grow. No growth of bacteria. So meaning that when you remove DNA from the mixture, the bacteria didn't grow and they cannot infect the mouse. Okay, so this is how every MA cloud prove that DNA is the factors, is the factors that cause the infections to the mouse. Okay, don't worry if you cannot understand fully this experiment. I will go one by one the experiment. Okay, now that we know at that time, they know that DNA is the one that will transform the harmless bacteria into pathogenic bacteria. But many other scientists are still skeptical because of the nature of the experiment, which is very basic. That time the experiment is considered very basic. So finally, SC and Chess, with the technology and everything and the discovery of DNA structure by Watson and Crick, so Hershey and Chess, they use another experiment, a quite similar experiment to the Avery, McCarthy and McLeod just now, but more, the technology they use is more advanced, so the information, the data is more believable. This is the Hershey's and Chess experiment.
Chase experiment was based on the biology of a virus called bacteriophage T2 that attacks a bacterium. The virus consists of a DNA core packed inside a protein coat. Part, but not all of the virus enters the bacterial cell. About 20 minutes later, the cell bursts, releasing dozens of particles that are virtually identical to the infecting virus particle. Hershey and Chase deduced that the entry of some viral component affects the genetic program of the host bacterial cell, transforming it into a bacteriophage factory. They set out to determine which part of the virus, DNA or protein, enters the bacterial cell. To trace the two components of the virus over its life cycle, Hershey and Chase labeled each component with a specific radioisotope. They used P32 to label DNA. The two strands of DNA have a sugar phosphate backbone that contains phosphorus atoms. Phosphorus is not present in most proteins. They used S35 to label proteins. Proteins contain some sulfur because sulfur is found in the amino acids cysteine and methionine. Sulfur is not present in DNA. After growing the virus in the presence of either P32 or S35, the two radioactive viral samples were added along with the host bacteria to fresh culture medium. The viruses were given enough time to infect the bacteria, after which the culture medium was agitated in a blender to detach the viruses from the bacterial cells. Samples were then placed in tubes and centrifuged to force the bacterial cells to the bottom of the tube, forming a pellet. The supernatant fluid contains the viruses. In each of these samples, the radioactive component will be in a different fraction, either the pellet or the supernatant. If DNA is in fact the genetic material, predict which of the two tubes, the one with P32 or the one with S35, will have a radioactive pellet. Hershey and Chase found that most of the P32 is in the pellet with the bacteria, while most of the S35 is in the supernatant fluid with the viruses. Because the P32 labeled the DNA, the scientists concluded that DNA, not protein, enters bacterial cells and directs the assembly of new viruses. That is, DNA is the genetic material.
Okay, so from the video, we know that the experiment, Hershey and Chase, they label virus. You know the structure of the virus, they have protein code and genetic materials. So they use two different labeling. One experiments, the batch one, here, they label the protein. Then the second experiment, they label the genetic material. Then they will allow the virus to infect the cells. This is how the COVID-19 infect our cells, the same mechanism. So the virus will inject their genetic material into the cells to infect the cells. So we want to know what is the genetic materials. So for the batch one, they label the protein. So either protein or DNA. The batch two, they label the DNA. So what happened is, after the infections of the virus into the bacteria, the centrifuge is a machine that will separate molecules based on their density. So bacteria, is larger than virus. So the bacteria should be settled at the bottom. Settle at the bottom here. So the first batch is labeled the protein. So if the protein is the one that get inside the bacteria, they should settle at the bottom because the bacteria is denser, lebih berat. Okay. But what happened is the label is observed on the supernatant, which is the less denser. So from the first experiment, that the protein is not transferred inside the bacteria. They stay outside, meaning the infections does not include the protein. From the batch 2 experiment, that only the DNA, only the genetic materials is transferred inside the cells, inside the bacterial cells. So meaning that when a virus infects bacteria, the genetic materials will get transferred inside the bacterial cells, leaving the protein code behind. So meaning that in order to transfer the virus genetic material, they need to insert DNA inside the bacterial cells so that the bacterial cells can multiply virus using their genetic material. I think if you read many articles about COVID-19, then you will understand how virus infects cells. Okay. So this is what Hershey and Chase do to prove that DNA, not the protein, not RNA, is the one that will infect cells, infect the mouse. Okay, so from these three experiments, the DNA was accepted as the genetic materials. And when the scientific community widely accept the DNA as the genetic materials, not RNA, not protein, not other macromolecules, so the challenge is to determine how the DNA structures functions, okay, related to their functions as inheritance factor, genes as genes. So far, okay? Don't worry, this one is just an introduction, just history and everything. So after this, we will move on to the main topics, the DNA replications. But before we move on, please read about these three topics. You can Google, you can refer to your textbooks, or you can go through my slides. So please read about 10 minutes reading times, the double helix, complementary strands, and Chargaff rules before we move on. Because there are some survey or experiment on students' learning behavior. If you do some 30 minutes reading time, so this one we make it short, 10 minutes. If the student do some 30 minutes reading time before lectures, your understanding will be better. So we will try the hypothesis, whether it's working on you or not. So please have 10 minutes reading time. Okay, we move on to the first topic. Yang tadi itu is quite important juga ya. The history. Sometimes I will put the history, so the experiment, especially the experiment in your final exam or midterm questions. Okay. Tapi don't worry if you cannot understand the experiment. So when we go through this chapter, you go back to the experiment whether you can understand or not. If you still cannot understand, then you can find me. You can text me, Telegram or everything. Okay. So the DNA structure. Now that we the DNA structure that we know is based on James Watson and Francis Crick. Their model, where they propose the DNA model, is very accurate. Okay, very accurate because they are based on these three very important informations at that time, 1950s. You know, the technology is still very not very advanced as compared to now. They don't even have a cell phone like you guys. Okay, so at that time, DNA is a non-molecules, but they don't know the DNA importance in our cells. They just know that this is DNA. Okay, inside our cells, there are DNA. Okay, and then Chargaff, a report on DNA-based compositions, which is the proposed Chargaff rules. Okay, we will look at the Chargaff rules later. And then the Maurice Wilkin and Rosalind Franklin. Rosalind Franklin is the well-known scientist, woman scientist. Okay. At that time, 1950, you know, the different genders, scientists is dominated by male scientists. Female scientists is very rare. So Rosalind Franklin is one of the most decorated female scientists. X-ray crystallography. This is what I'm doing right now, actually. X-ray crystallography is still people are using this technology until now, X-ray crystallography, but not for DNA molecules, but more to proteins, individual proteins. Okay, first we look at the Chargaff. So Chargaff rules. Chargaff proposes Chargaff rules, which is the compositions of DNA varies between species. So DNA, they have diversities. Okay, so each person's your DNA is different from your friends, from your parents, except for identical twin. Identical twin, you will have the same DNA compositions. But other than that, even between insects, between fishes, between birds, they have different DNA compositions. So that is the evidence that the DNA can be the candidate for genetic materials. Because in order to be a genetic materials, the materials need to be diverse, okay, different. The molecule is the same, but inside the molecules, there are some diversity, which is the genes. Different people, they have different genes. That's why we have different skin color, different appearance, different characteristic, right? Because of the diversity of DNA. And Chargaff rules stated that the number of bases are equals. A equals to T, G, and C. If you read about complementary strand just now, or Chargaff rules, you will know. So DNA have 4 words: A, G, C, T. I always write the A, G, C, T like that. Like this. It's easier for us to memorize. Okay. A, G, C, T. So G will always pair with C, A will always pair with T. So I always write this, the bases, DNA bases like that. Okay, that's why A equals to T, because A will always pair with T. G equals to C because G will always pair with C. Okay. Okay, that one is Sargaf rules. Second one is based on X-ray diffractions photograph of DNA. X-ray crystallography, they will actually an experiment where they will expose the molecules, in this case DNA, they will expose the DNA molecules with X-ray. You know what is X-ray, right? Because you have X-ray of your lungs, you have X-ray of your body. So in this case, they will expose the DNA molecule with X-ray. So the X-ray will bombard the DNA molecules and they will produce a film. Okay? Like your X-ray of your body. You will get a piece of paper, not paper, like a film like this. So the molecules, they will show the helix, the nitrogenous bases, and everything. The patterns, we call this diffraction pattern. So from the diffraction pattern, you know the molecules. You can use software from computers to deduce the molecules inside the DNA. So this is how Watson and Crick built their model based on these X-ray diffractions. And if you read more about Rosalind Franklin, actually the experiment at that time, they don't know the side effect of X-ray. So it's not a good ending for Rosalind Franklin actually. Okay, so from all that, Watson and Crick built a double helix. This is when they are still young. Now they are very rich now. But I don't know if they're still alive. Okay, so Watson and Crick built a model. They proposed a model of double helix and anti-parallel to each other. One run in the 5-3 directions, the other one run in the 3-5 directions. Okay, this is a 3D models of their DNA structure. And DNA composed of 4 nucleotides, which we discussed just now from Chargaff rules: A, G, C, and T. Adenine, cytosine, thymine, and guanine. And this is the Chargaff rules: A, T, G, C. A and G is purines because they have 2 rings. G and T and C is pyrimidines because they have 3 rings. This one is chemistry. Okay, based on the structure, Watson and Crick stated that DNA is a double helix, double-stranded. So they have 2 strands anti-parallel to each other. 2 identical strands, but only they are anti-parallel to each other. The directions is different. Okay, dia molekul yang sama, 2 strand, tapi dia terbalik. Okay? Susunan dia just terbalik sama satu sama lain. Tapi actually dia sama. Okay. Nucleotides, the AGCT is linked by 5' to 3' phosphodiester bond. This one is chemistry, you know. I mean, like the peptide bonds, everything. This one is phosphodiester bond. And then the 2 strand is linked together by hydrogen bonding between the A and T and G and C. The AGCT is a hydrogen bond, but the strand di sini, they are linked by phosphodiester bond. But between the 2 strands, di tengah ni, They are linked together by hydrogen bonding. Okay, like this one. This is the first strand linked together by phosphodiester bond. Second strand, same, linked together by phosphodiester bond. But between the two strands, GC and AT, they are linked together by hydrogen bonding. Here. TGCA, they are linked by phosphodiester bond. Okay. T, G, C, and A. This is one strand. One strand. So they are linked together by this one, phosphodiester bond. But between the two strands, first strand, second strand, they are similar. Only the direction is different. One is 5 to 3. One is anti-parallel to each other. Actually, they are same. T, T, G, G, C, C, A, A. Yeah, the two strand is linked by hydrogen bond. Yeah, between A and T and G and C, hydrogen bonding. So there are two bonds. Hydrogen bond between the two strand, phosphodiester bond within the strand. Okay, boleh kan? Okay, this is purines. They have two rings. Adenine and guanine, two rings. Cytosines, they have only one ring, not three rings. Okay, my mistake just now. Thymine and cytosine is pyrimidines. Okay, this one is detailed explanations of the Chargaff rules. Okay, skip this one. Okay, I think this one you already learned in your first semester. Nitrogenous base, nucleotides. Nucleotide is this one. Oops, sorry. Nucleotide. Nucleotides composed of is the building block. Building block. Building block of DNA is nucleotide. Yeah. Like peptide, they have polypeptide, they have their own building block. Carbohydrates, they have their own building block. Right? Okay, we have another video. This is a difference between DNA and RNA.
DNA and RNA are nucleic acids, polymers made of. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxy. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay. Okay, so that is the DNA structure. Okay, we move on to the DNA replications. So now we know DNA structure is double helix and antiparallel, opposite directions. So DNA must be able to make copies of itself. So DNA should be able to copy, because we know that inheritance, you requires to copy everything from one cells to another cells. Like how we inherit something from our parents is by copying our parents' genetic materials, but not exactly. However, in our cells, because we always renew our cells, each of our cells, they have a similar DNA. That's why when they at the crime scenes, they take your blood sample, they take your hair samples, they can trace your DNA, because each cells in your body have the same DNA compositions. Okay? So that's why when you're still little, you divide your cells, is dividing and everything. They come from single cells. You come from single cells. Yeah? From sperm to ovum, and then you have your own DNA mixed between your mother and father. And then you have your own DNA. Your DNA will make copies of itself into the cells and everything, and you will become a multicellular organism like now. So that's why your DNA in each of your cells is the same, except for your gametes. Okay. Okay, so based on the DNA model, we know that DNA is complementary. Means each strands of DNA is the same, only it runs in the opposite directions. So each of the strands can serve as a template for building new copies. So in DNA replication, the molecules will separate, unwind, and the new strands are built based on base pairing rules, Chargaff rules, AGCT. So that's why the Chargaff rules is very important. So the replications of DNA can be explained by using a semi-conservative model. Because before the semi-conservative model is being confirmed, there are other models being proposed. Actually, there are 3 models being proposed here. The conservative and dispersive model. But because of the experiment by Michelson and Stahl, this one you can read, because I already give you idea why I include many experiments in my slides. Because to understand, in order for you to understand that everything that we learn is a result from several experiments, is proven by experiment. In science, in biology, we cannot say that vaccine is safe, and then you believe that the vaccine is safe. No, we need to give proof. How we prove the vaccine is safe? Through experimentations. So this is why we learn something because of experimentations. Kalau kamu mengadu, banyaknya kena baca biologi ni, banyak kena belajar. Because of these people lah, they do many experiments. So because of the experiment, we have to learn many things, theory. But it's for good things lah. Without the experiment, we cannot understand about DNA, so we cannot understand the virus, we cannot produce vaccines. So everything happen for a reason. Jadi belajar jalan. Okay, so the semi-conservative model is proven where original double helix DNA will unzip. Unzip kita lebih sesuai guna zip lah, because later on we will look at the replications punya mechanism. Actually, it's quite similar to the zip. Your zip. Kalau kamu ada zip, kalau pakai seluar ada zip, pakai jacket ada zip. Okay, so the semi-conservative model. The other model is cancelled because of Michelson and Stahl experiment. They proved the semi-conservative model. This one kamu boleh baca lah. Okay, so Watson and Crick semi-conservative model. Parent molecules will separate. Okay, so each of the strand they act as a template for replications. So dalam satu cycle of DNA replications, we will produce two DNA molecules, daughter strand lah. Okay, so this one jadi template. Dia akan buat baru based on base pairing rules, Chargaff rules. AT, CG, AT, AT, GC. So based on this one. Same with the other string. Okay, if you ask where the CTAG come from, they always produce in our cells from what we eat. So we can have our own AGCT, nucleotides inside our cells based on food that we eat. Ini kita skip lah. If you have any questions about this experiment, you can ask. I think this one you will discuss in tutorial juga. Okay, the mechanism of DNA replications. Okay, first we look at the enzymes and proteins that participate in DNA replications. There are many. Actually, this is the most important one. Helicase. Helicase is the enzyme that will unzip the double helix. Binding proteins is to stabilize separate strand, because your double helix is twisted. Dia bergulung-gulung. Your DNA is twisted like in chromosomes. So binding protein is the one that will stabilize the strands. Dia jadi straighten lah, macam rebonding. Dia jadi straight. Primase is the enzyme that you add short primer oligonucleotide to template strand. Because DNA, different from mRNA and RNA, they cannot be created from nothing. They need to start at primer. So primers add primer, so the DNA can be replicated. DNA polymerase III is the most important. Bind nucleotides to form new strand. DNA polymerase III is one yang akan cari nucleotides. Questions? Okay, this one relax. We just started ya. Jadi kita start. This one enzyme. DNA polymerase I, exonuclease, remove RNA primer. Okay, the primer tadi is added by primase. but later on will be removed. Okay, because we don't want RNA, we want DNA. Okay, DNA ligase. This one is for lagging strand, join a consecutive fragment. Seals adenine in sugar phosphate backbone. This one will seal DNA ligase. Because the DNA replications, one strand is straightforward. The other strand is, the antiparallel strand is a bit complicated. But if you understand the concept, it is easier for you to understand. Okay, DNA replications, they begin at origins of replications. Jadi dia start bila ada origin of replications. Meaning that there are several protein that trigger. Meaning maybe kita perlukan insulin. Jadi some of our cells machinery mechanism, they will trigger the origin of replications for the insulin genes.
The DNA molecule begins with a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of the eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
Okay, really there are slight difference for prokaryotic and eukaryotic. Tapi most of the content more towards eukaryotic because we are eukaryotes. Okay, this is the origins of replication just now. Dia akan macam creates bubbles. This one is for prokaryotes, so very straightforward. Prokaryotes, most of them is circular DNA, but for us is double-stranded. Okay, this one. So compared to prokaryotes, eukaryotic cells, they have many origins of replication. So the replications of DNA, they occur at the same time, both directions. Okay. So at the end, they will have two daughter DNA molecules. Yeah, this is the process. Video dulu, then baru kita tengok satu persatu. Okay. Very good questions yang tadi.
Is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T.
Ini represent genes lah.
Strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called replicase and results in the formation of a replication fork.
Macam zip kan?
The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process.
Primase add RNA primer.
This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer.
Okay, this one yang lagging strand.
DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then
fragment
made,
by
and the
fragment.
process is repeated
So
again.
the fragment is called
Once the
Okazaki
new
fragment.
DNA has been made, the enzyme exonuclease removes all the RNA primers from both
RNA
strands of
kena
DNA.
remove sebab kita mau
Another DNA
DNA.
polymerase enzyme then
Dia
fills in
tukar
the
dengan
gaps
DNA.
that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Semi-conservative. Okay, boleh? Start. Before we go by one. Tapi I will go faster lah because we already have the video to explain everything. Okay, in the video you can see that the unzipping will create in the origins of replications, they will create a replication fork. Kita panggil dia tadi kan, dia ada dalam video tu. So replication fork macam zip lah, Y shape. So where new DNA strands are elongating. So the unwinds or the unzipping is because of the helicase. This enzyme. Helicase. So helicase move in this direction to unzip the double helix. Jadi dia terbahagi dua. Jadi dia boleh jadi template lah. Template 1, template 2. So one template dia akan jadi leading continuous. The second template is lagging. So dia fragment by fragment. Jadi fragment tu kita panggil Okazaki fragment. Okazaki is the scientist that discovered this mechanism. Japanese. Okay, this is the enzyme topoisomerase that stabilize the unwind of the DNA strand. Tadi dia twisted tu kan, dia macam dia luruskan. And then this one single-stranded binding protein stabilize the single strand supaya dia tidak bergulung-gulung gitu. Dia maintain macam ni supaya ada replications process can be a bit easier. Okay, for replications, the most important enzyme is DNA polymerase 3 actually. DNA polymerase and RNA primase enzyme. Because without primer, DNA polymerase cannot synthesize new DNA. Because DNA cannot be created by things. So the RNA primer serves as the starting point. Starting point for DNA synthesis. Okay, so you can see here leading strand is continuous from 5 to 3 direction, continuous. But for the other strand, the lagging strand, fragment by fragment, the direction same here, tapi dia add di sini dulu. Fragment 1, lepas itu dia add lagi, fragment 2, add lagi sini, fragment 3. Later on, the fragment, 3 fragment will join together by DNA ligase. That one is for the lagging strand. Okay, this one sudah tengok dalam video kan. So new nucleotides will be added into the 3N. Jadi the direction is from 5' to 3'. This one is very important that you have basics chemistry 5' to 3'. So 3' ada OH kan. Jadi nucleotide triphosphate dia akan undergo reactions that will release the triphosphate into pyrophosphate. Jadi tinggal satu phosphate. Jadi that's why kita panggil dia phosphodiester linkage. Dia jadi covalent bond. But this one is more chemistry. You don't have to explain the chemistry part. Okay, another. So far, okay, the leading lagging strand. Kalau okay, we don't have to play this video. Play aja lah kan. Sekejap je.
Simultaneous formation of the leading and lagging strand. It's a continuous leading strand while primase, other DNA polymerases, and
Yang
ligands
bawah tu
all work
leading.
together to make the
So
lagging
dia
strand.
continuous.
The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Okay, leading strand.
Using more
Continuous.
Here you
Okay. This one is for continuous. So they add primer, RNA primer, then bind di sini. Ini akan continue sampai ujung. Tapi untuk lagging strand,
the other new strand is called the lagging strand.
lagging strand a bit different.
The lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5'.
Jadi lagging strand dia ada banyak primer lah. Dia add satu-satu. Then after that they remove the primer.
Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand.
Okay, and then joined together by DNA ligase.
Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding protein, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase
links the Okazaki fragments to form the lagging strand. The other new strand is called the lagging strand.
Okay, so far okay. This one, if you don't like video, you can use this one. Okay, kalau the leading strand, they're straightforward. But for the lagging strand, RNA primers synthesize primer at different locations. This one is first fragment, the other their own primer. Then the DNA polymerase will add DNA, yang biru to DNA. Then the primase, they add another primer. They add another primer, and then the DNA polymerase will synthesize again the second fragment. Then the two fragment will be joined together. First, they remove the RNA primer yang merah tuh, because we only want DNA. Then they join together by DNA ligase. Okay. That's for the RNA replications. It's very complicated process, but when you get the concept, actually it's very important. So what you need to focus is the complementary strand, leading strand, lagging strand, and all the enzymes. Okay, the DNA replications model, they have two problems: the N replication problems and proofreading. So N replications problem is because when the DNA is because of the lagging strand. Lagging strands, they can only remove this one, remove the RNA primer, and then DNA polymerase will replace the primer, RNA primer, with DNA. But at the end, they cannot, because they can only add to the 3N. This one is the 5N. Kan, sini 5N, so they cannot add DNA polymerase. Here they can. Di sini pun boleh, di sini pun boleh. Tapi di ujung sini they cannot add. They can remove the primer. They can remove this, but they cannot add DNA because this one is 5 prime. DNA can only be added to 3 prime. Right? Nanti kamu tengok balik what I just explained. Yang slide nih. Okay. So this is the problem, the lagging strand. Jadi di ujung lagging strand tuh dia jadi empty ini. Jadi in order to avoid that, they just remove the other strand. Ini dia remove lah. Jadi the DNA is complete DNA replications. So the problem is N replication problem. When this happen, each round of replications will make our DNA shorter and shorter because of the N replication problem. That's why when we get older, we lose a bit of, kita jadi macam less functional because our DNA semakin kita tua dia semakin short, kan? Jadi mungkin kita kehilangan beberapa genes lah. That's why when we get older, we become less functional. And actually there are some mechanism or enzyme that is responsible for the to minimize the effect of shorter DNA, untuk meminimumkan efek kita punya DNA replications yang memendekkan kita punya DNA itulah. So telomerase enzyme, the functions is to add repeats unit at the 3 overhang yang tadi itu. Instead of removing yang the hanging tadi, that we cannot add any more DNA di ujung Okazaki fragment tu, the telomerase will repeat this one. They repeat. So dia akan panjangkan lah this one. So each replications kita dapat avoid the shortening of our DNA. And actually the telomerase enzyme has been the subject, the focus of pharmaceutical company or other company to prolong our life. Jadi kita boleh hidup lebih lama lah when the research about telomeres is complete ataupun dia bagi hasil. But maybe you can read more about this. So the end replications problem, the solutions is telomerase enzyme. Another problem is proofreading and repairing of DNA. Okay, during the process of DNA replications, the DNA replications will be monitored. Dia akan, inilah dia punya quality. But then, the DNA replications is not perfect. It is very efficient, but it's not perfect. Sometimes there are mismatch of DNA pairings of the Chaga rules. Sometimes AT dia jadi AA, AG gitu. And then in addition to that, DNA can also be damaged by exposure to chemicals or physical agents. For example, X-ray, like what happened to Rosalind Franklin. Because at that time they don't know the effect of X-ray to our DNA. So that's why Rosalind Franklin, during the experimentations, they are exposed to X-ray for a very long time. So that caused cancer. Yeah. Okay, so X-ray is one of the example that can cause a damage to our DNA. So our DNA replication system, actually they have proofreading and repairing system. So after each DNA replication cycles, your DNA will be proofread. Okay, will be proofread and repaired. For example, here when your DNA is being damaged by X-rays, the nucleus will remove remove the damaged DNA, and DNA polymerase will repair. DNA ligase will join back the new DNA, because this one need covalent bond. Covalent bond need to use DNA ligase. Okay, this one is very short. Maybe you will discuss more in your tutorial sessions. So we will have another 10 minutes reading time. Atau short break juga lah this one. 10 minutes reading time. Please read about this messenger RNA, codon, RNA polymerase. You can Google, you can refer to your textbook. Okay. So for the last topic, protein synthesis. Protein synthesis is when we need to express. So that's why kita panggil the expressions of biological informations. Bila kita need to express the genes. Genes tadi kan dia huruf A, G, C, T, C, G. Susunan dia itu actually is translated. Kita boleh transcribe, kita boleh translate into something. Normally protein lah. Okay. Jadi informations dalam DNA itu, atau kita panggil genes, dia ada informations. Dia ada directions untuk buat protein. Dia punya simple term lah. Ada eksperimen juga. Bidal-lentatum. This one kamu boleh baca. One gene with one polypeptide. So bidal-lentatum is the one that discover this one gene for one polypeptide. Jadi satu genes dia mewakili satu protein. Macam dalam your last lecture last week, Mendel law of inheritance. Macam kacang pis tu dia bulat atau wrinkle kan? Round or wrinkle. So that one can be one genes, one polypeptide. Can be related to bidal-lentatum experiment lah. Okay, yang tadi tu gene expression kita panggil ya. When you want to use the information inside DNA to make protein, that process is called genes expressions and involve two stages, which is transcriptions and translations. Transcriptions is when the DNA is transcribed into mRNA. mRNA ini dia ada codon. And then mRNA will be translated to ribosome. mRNA ini messenger RNA. Sudah baca kan? So dia just jadi messenger lah. Dia bawa the information to ribosome. Ribosome ini kilang. Kilang untuk buat protein. Dia bawa the information, dia bagi pada ribosome. Ribosome akan translate the information into protein molecules. Okay. Bila dalam DNA, information itu dia means nothing. Dia just huruf lah, A, G, C, T, C, G, C, T itu. Tapi bila dia pergi ribosome, dia jadi protein. Protein itu yang akan buat kita ada karakteristik. Macam skin color is because of protein. Eye color is because of protein. Kita punya suara pun because of protein. So, whenever the DNA is being transcribed and translated, that's when you show your characteristic. So that's why now kita ada gene editing kan, DNA. Kita boleh edit kita punya DNA. Kita panggil dia CRISPR-Cas system. Even vaccines pun actually is modified by the same concept. Okay, kita create the virus punya mRNA. So bila kita masukkan vaksin mRNA itu dalam badan kita, mRNA itu akan use kita punya cells machinery to produce virus itu punya protein. Tapi protein itu tidaklah bahaya sampai dia infect kita. Just to trigger our antibody defense system. Okay, the gene expressions from DNA to RNA and protein, mRNA to protein actually is kita panggil dia central dogma of molecular biology. Okay, for my postgraduate study actually, my degree is biotech. So this is what we learn in my 3 years, including my postgraduate, central dogma of molecular biology. So you will always play with DNA, you will always play with protein. So this concept is being discussed every day if you want to take biotech later on. UMS ada biotech juga, if you're interested. Okay, so transcription is the synthesis of RNA ataupun messenger RNA under the directions of DNA. Directions mean dia punya informations from DNA lah. Dia akan menentukan messenger RNA itu macam mana dalam bentuk codon. Okay, RNA dia akan travel dia akan cari ribosom. So bila dia jumpa ribosomes, the ribosomes will translate the informations from RNA tadi into protein. So this protein yang akan bagi kita karakteristik ataupun enzim macam insulin gitu. Okay, this is the process. Harus cepat je lah ya. Okay. Why we need mRNA? Sebab in eukaryotes, our DNA is too large. They cannot pass through the nucleus. And also, DNA should be enclosed within nucleus. Kalau DNA kita keluar, bahaya. Sebab DNA is the one that store all our genetic informations. Jadi, in order to avoid that, the DNA is transcribed first into mRNA. mRNA yang akan travel outside the nucleus, dia akan cari ribosome lah. So, in this way, DNA is conserved, dia dipelihara, dia dijaga lah dalam nucleus. And then at the same time, the informations inside your DNA can be translated by using mRNA. Okay, before we discuss about transcriptions and translations, you need to understand the genetic code. Genetic code is based on triplet code. Triplet code. Dan benda ni nanti dia akan jadi codon. Kita panggil dia codon. Okay, template strand. Only one of the two DNA strands dia become template. Kalau DNA replications, they use both strand. But for transcriptions, only one strand. Okay, so codon is the triplet code that being used by mRNA from DNA. Okay, inilah begini dia punya triplet code. So in DNA kita ada AGCT. For mRNA, macam tadi dalam video tu dia explain the difference between RNA and DNA kan. So the difference is the T is replaced by U in mRNA. Jadi kalau T tadi dia replace jadi U lah. G, T jadi U, T jadi U. G, C jadi U, C, A. So template dia is this one. But this one is complementary to this one. Yang ini bukan template nih. Okay. Only one strand is used as template during the transcriptions, as compared to DNA replications where both strand become templates. For DNA transcriptions, only one strand. Jadi kita tengok nih, dia complementary dengan mana? So strand yang inilah. Yeah. Okay, during translations, the triplet code will be translated into amino acids. So amino acids, they combine together, they will become protein or polypeptide. Okay, so what is codon? Codon you can use to this table here. Triplet codon. So there are total 64 codons, 61 code for amino acids, and 3 codons are for stop. This stop codon. UAG, UAA, UGA. Kalau kami memorize dia WAG, WA, UGA. And start codon AUG. They will code for methionine. Okay, each codon they can code for the same amino acids. For example, like this one, CGU, CGC, CGA, CGG, they code for the same amino acid, arginine. Same with the AGA, AGG, arginine. Jadi kita ada total 20 amino acid. Jadi this 64 codon, some of them code for the same amino acid. Okay? Like this one code for the same amino acid. 64 codon, but only code 20 amino acid. So that is the codon. kita panggil dia redundant sebab different codon can code for the same amino acid. Genetic code is universal, meaning that if you put your DNA inside E. coli, for example, the E. coli will produce the same protein that you produce inside your body. That is genetic engineering. Nanti kita akan tengoklah the gene tech punya chapter, macam mana kita boleh transfer genes from human to bacteria, and the bacteria will produce the same protein that the human produce. So that's why we call them universal. Promoter.
An enzyme called RNA polymerase carries out
RNA polymerase, they bind to the
with
promoter.
the help of protein transcription factors. It attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation.
Okay, only one strand become template. Sebab dia cuma perlukan satu strand.
Template for
Kalau
RNA.
DNA replications, dia perlu dua.
RNA polymerase moves along the DNA strand in the 3'
This one
5'
elongation.
direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
T dia ganti dengan U.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Okay, this one is termination. Terminations when the RNA polymerase encounter terminator sequence. Okay. Ini sama macam video tadi itulah. Initiation, kita ada promoter. So, promoter ni dia punya karakteristik ialah dia ada tata box. So transcription factors dia bind, and then RNA polymerase dia akan bind with the help of transcriptions initiations factor. So dia bind, dia akan start lah transcribing. Okay, RNA polymerase, RNA can be created from without primer. So dia tidak perlu primer. Dia direct transcribe. So based on the base pairing. Cuma beza dia T is replaced by U because the codon for the mRNA is different ya. Tapi the concept is the same. Same with the DNA replication. Okay, this one is elongation. And finally terminations. DNA polymerase detach when they reach atau they read terminator sequence. Maksudnya hujung genes itulah. Genes dia ada promoter, dia ada terminator juga. So depends on the genes. The RNA polymerase is a very smart enzyme. They can detect that. Okay. Okay, boleh kan? Transcriptions, initiations, elongations, terminations. Very simple actually. Kalau kamu sudah paham konsep DNA replications, this one is very simple. Okay, for eukaryotes, kita ada exon, intron, and exons. Kalau prokaryotes, dia tiada introns. Introns tu actually, they are introns in our DNA because to conserve, to minimize the effect of mutations. You know, mutations, kadang-kadang dia akan ubah kita punya genetic sequence. Tapi bila kita ada introns, kadang-kadang the mutations occur in the introns. Jadi introns is non-coding regions. Jadi kalau dia ubah pun, no effect. Kita panggil dia nonsense mutations. Because the mutations occur in the non-coding regions. So nanti kita expand lagi lah the next chapter, maybe chapter 4 or chapter 5, ada mutations. Gene tag mungkin. Jadi dia buang intron tuh before being transferred to ribosome. The mRNA will remove introns. Kita panggil dia splicing lah, RNA splicing, remove introns and join exons. Because only exons is coding regions. Yang ada information untuk buat protein cuma exons. Jadi kita kekalkan exon, barulah kita pindahkan dia pergi ribosom. Okay, sudah proses RNA splicing. So translation. Translations is a bit longer. Boleh lagi kan? Saya ambil masa kamu. You don't have any class kan? For 30 minutes. Your next class will start at 10:30 kan? Okay, sorry I take a bit of your time today. Okay, translation. Translations is when you have the messenger RNA sudah. The translations, the messenger RNA will be transferred to ribosome. And then with the help of transfer RNA, tRNA. So ada 3 lah benda yang terlibat di sini, mRNA, tRNA, and ribosome. Kita tengok dulu the structure of tRNA. tRNA, they have amino acid here. Okay, they have amino acid attachment. Jadi different tRNA, different anti-codon. Sebab di mRNA kita ada codon kan. So the codon is the complementary to the anticodon here. So AAG dia punya codon is UC lah. AUGC kan. So codon is, mRNA codon dia is C. Bukan T ya, T untuk DNA. Okay, so AAG dia punya amino acid. Kita boleh refer to the table tadi. Tengok dulu. Bukan AAG. UUC. Codonnya. Jangan refer to the anticodon. Kita refer to the codon. UUC. So, phenylalanine lah. Dia punya. Boleh kan? UUC. So, phenylalanine. Codon dia UUC, jadi dia punya ni is UUC lah. So, okay, refer to codon table yang ada 64 codon tu. This is the 3D. Jadi kita akan guna this structure lah untuk explain everything. AAG anticodon dia. So the C. Jadi di sini dah. Ini. Boleh kan? That's how you find use the table. Jangan refer yang ini. Yang AAG ni, not this one. Ini anticodon. Kita refer yang ini. mRNA dia. Okay, ribosome. Ribosomes, there are 3 sites. E site, PE site, A site. The different sites, they have different attachment. Jadi, for the initiations, E sites. So, ini a growing polypeptide. Yang A site tu P kan? So, P, polypeptide. Di A site dia amino acid. Dia akan bawa satu amino acid. Itu yang growing polypeptide dari P site tu dia akan pindah ke A site. Okay, kita tengok initiations, elongations, and termination. During initiations, mRNA akan bind to the small ribosome subunit. Ribosome kamu sudah belajar kan? Dia ada small subunit, dia ada large subunit. Okay. Small subunit, large subunit. Okay. Translations pun sama juga macam transcriptions. Dia ada 3 stages, initiations, elongations, and terminations. Kita pergi ke initiations dulu. First initiations, what happen is the mRNA will bind to the small ribosome subunit, and then they will scan They will scan until they find the start codon. Dia scan sampai dia jumpa start codon. What is the start codon? AUG. Jadi start codon dia akan cari antikodon dia lah. Antikodon di RNA yang ada antikodon UAC. Di UAC dia methionine. Ya, nanti kamu refer balik sama kan? The table, codon table tadi. Oke. Bila dia sudah jumpa start codon, the large ribosomal subunit akan datang bind. Yeah. So this is the initiation stage. Elongations. This one initiations lah. Animation. Okay, first dia cari dulu the start codon. Bila dapat start codon, the tRNA punya antikodon akan bind. Methionine lah, dia buat methionine. Then large ribosomal subunit akan bind. Okay, elongation. Elongations, amino acid is added one by one di A site. Okay. So the second codon tu apa? Dia akan baca lah. So dia akan cari antikodon. Okay. Based on the information sini. Based on the codon di A site. So growing polypeptide akan pindah di sini. Peptide bond formations. Dia ada 3 step. Codon recognitions, dia scan what's the codon and then they find the suitable tRNA anticodon. Then peptide bond formations from the growing polypeptide at the P site tadi, P site pindah ke A site. Okay. Jadi this one is number 1, number 2, number 3, number 4. Yang ini nanti akan jadi kelima lah, amino acid yang kelima. Okay, so 1, 2. Okay, pindah. Okay, translocation. Bila dia sudah pindah, the mRNA will move. Will move to this direction. Jadi yang E site tu dia sudah hilang kan. Polypeptide sudah pindah di the next tRNA. So dia akan remove lah. And then dia akan cari lagi the next tRNA. Based on the codon. Okay, this is the animation lah. Start codon dia pindah pergi the A site punya peptide bond formations. And then bergeraklah. The mRNA will move towards the E site. Yang the P site tadi tu pindah ke E site dan dia keluar. Jadi dia start again and again and again and again. Hopefully okay ya. Kalau saya pelan buat cepat. Nanti next week saya explain lagi lah. Okay, terminations. Same thing with the transcriptions. Tapi untuk this one, terminator sequence dia kita tahu. Kita ada 3 stop codon. Whenever the messenger RNA, they scan for UAG, UAA, or UGA, UGA, stop codon, dia akan bind release factor, not tRNA anymore. So, bila dia jumpa stop codon, instead of tRNA, dia akan cari release factor. So release factor, they can causes additions of water. So water process hydrolyze kan? Hydrolysis. Jadi bila ada hydrolysis, the polypeptide will be removed from here. Jadi complete. The polypeptide complete. They become protein. Maybe this protein is insulin. Ataupun any protein. It can be any protein. Depends on the informations from your codon. Codon that is being transcribed from DNA. Simple. Central dogma. from DNA to mRNA, from mRNA to protein. Two process, transcription, translation. And then transcriptions, dia ada 3, initiations, elongations, terminations. Same thing, translation, initiations, elongations, terminations.